Dear students, I am Dr. Aruna Mohan from Delhi University. I will continue discussing on alimentary canal. In previous sessions, we have discussed about buccal cavity, secretions, about stomach, the secretions from stomach, about the duodenum, and also about the accessory glands of digestive system, to name pancreas and the liver. We know the juices of pancreas and the bile from liver will drop inside the duodenal cavity. And also we are aware of the fact that fats can be digested only after emulsification of fat and activation of fat digesting enzyme lipases. In this session, we are going to discuss in detail about the intestine, small intestine, which is a very long coiled tube. Also, we will try to know what is happening to the food, the digestion and the enzyme when the food has reached intestine. Children, you are aware that some digestion took place in stomach. Before that, some digestion had taken place in cavity also. Then digestion continued in duodenum and now it will continue in small intestine. Small intestine is a very typical area of alimentary canal. So many important things are going to happen when food is in the small intestine. To start with, the goblet cells in the small intestine which will secrete mucus. Dear students, by now you are in a position to appreciate the secretion of mucose, the secretion of mucus in the alimentary canal, because it is going to protect internal lining of alimentary tract. So, goblet cells are secreting mucus. This is one thing. Another thing, the brush border cells in the small intestine will secrete a juice. Now, that juice along with the mucus is called succus entericus. And it is interesting to note that succus entericus is a common juice which contains so many kinds of capabilities. It has enzymes for all kinds of food, starting with maltases, dipeptidases, lipases and nucleosidases. What I mean by this? If I say maltases, means it is going to digest carbohydrates. Similarly, dipeptidases, it is going to digest proteins, lipases for fats and nucleosidases for nucleic acids. In other words, I am trying to emphasize that digestion of all these items has started. Digestion of all these items started right in the buccal cavity. Some digestion did take place in stomach, then in the duodenum, but again one more time we have availability of all these enzymes in small intestine in the form of succus entericus. What is the purpose? The purpose is that after digestion has almost taken place, whatever is left to be digested, the final finish will take place in the small intestine by succus entericus. So, our system is so well structured that anything which we eat should be digested, because we cannot absorb it unless we digest it. Digestion means breaking bigger molecules to the small, then again smaller, then smallest. And when it reaches to the final smallest stage, then only you can absorb it. So, alimentary canal is not leaving any chance. Whatever final thing is there, not digested so far, will be acted upon by succus entericus, and final digestion of the things will take place here, and that is why one juice contains capability of all other enzymes which you find in different parts in the alimentary canal otherwise. Other point which I would like to highlight, my dear students, that you know pancreas is secreting pancreatic juices which are going to help in digestion, which we have already discussed. In addition to pancreatic digestive juices, pancreas also secretes mucus and bicarbonates. Again, you already know the value of mucus in the alimentary canal, and bicarbonates which are getting mixed with the food now, which will turn acidic medium to alkaline medium. pH will come down to alkaline pH 7.8, and please remember the pH in the stomach was around 1.8, very acidic because of presence of HCl. So, now at this point I want you to realize and appreciate the point that the acidic pH of stomach, the food which is in stomach having acidic pH, is now moving to duodenum and then to small intestine, and pancreatic juices and small intestine succus entericus juices can act on this food only if medium is alkaline and not acidic, otherwise enzymes won't be able to act. So, the medium should be brought down to alkaline medium, and that is done by bicarbonates and mucus produced by pancreas. Again, it is very important step. You are bringing pH from 1.8 to 7.8, a very big change. So, there is alkaline medium after food is going from duodenum to small intestine. This mucus will also protect the intestinal mucosa. You know that when food is entering the duodenum, it is acidic in nature. It will become alkaline after receiving pancreatic juices and mucus and bicarbonate from pancreas. It will take some time, and by the time food is already entering the small intestine. So, this mucus is protecting intestinal mucosa or intestinal lining from acid, of course, and alkaline medium is provided and pH is brought down to 7.8, which is nicely alkaline medium. So, children, see how in our alimentary canal every step is so well designed, every secretion comes on time, neither before time not after time. Suppose it was not coming on time, digestion will not take place, and if it was coming after time, then pH will not change, and hence again digestion will not take place. Perhaps you already know that enzymes can act only at a particular optimum temperature and particular optimum pH. Hence, the maintenance of pH in different parts of alimentary canal becomes so important, and we have specified very specific, very special secretions in each and every part of intestine which does this important work for us. The proteins which we eat are broken down to its simpler forms like peptones, proteases, and now they will be acted upon by trypsin, chymotrypsin, carboxypeptidases to convert it into dipeptides. So, if proteins, peptones, proteases are converted to dipeptides, that means the molecule size has become further small, and that is the purpose. Digestion means we break it into its raw form, that is our effort, that is the effort of enzymatic action. Of course, you already know from where we got these enzymes. Coming to amylases, you have pancreatic amylase at this point of time. When food is in the small intestine, you have amylases from two sources. One is pancreatic amylase, second is amylases of succus entericus, which is the juice from small intestine. Now, polysaccharides like starch will be converted to disaccharides. So, poly means many, di means two. Finally, it will come to one. and that will be the absorbable form. Here I would like to remind you one more thing, that succus entericus, which is having combined action of all the enzymes, all kinds of food can be digested by this. Why succus entericus has that capability? Because the idea is to convert the complex food into its simplest form, not only simplest form, the form in which it can be absorbed. Students, please do not forget the fact, the food which we eat through our mouth should be digested and should be brought to the stage in which it can be absorbed, and then this absorbed food should reach the correct part of your body where it should be assimilated, and only after assimilation has taken place, then this food is going to give you energy, or this food will do the job for which you have eaten this food. So, eating is important, but digestion, absorption, assimilation, unless these three things also happen in your body, then purpose of eating food is not served, and that purpose is served by churning action, by action of enzymes, and then by the process of absorption. Assimilation comes later. So, we have converted polysaccharides into disaccharides in the presence of amylases. The origin in this part of alimentary canal will be pancreas and the small intestine. Coming to fats. Fats digestion has begun, emulsification has taken place, activation of lipase has also taken place. Now, this lipase is acting on the fat molecule and digesting it. Again, you have lipases at this point of time in the small intestine from two sources. One is from pancreas, the pancreatic lipase, other from the succus entericus, and you have bile also, which is activating lipase to act on fat molecule. So, these fats will be converted to disaccharides, that is from many to 2, and from disaccharides to monoglycerides. I would like to repeat this point. Lipases will act on fat, will convert fat into diglycerides, and these diglycerides will be converted to monoglycerides. That means it is becoming simpler and simpler. Of course, when it comes to mono, that is the simplest form. Now, it is ready to be absorbed. When it comes to absorption, again there is a point to remember. Proteins you have converted to its simplest form, amino acids, which will be absorbed by the blood. Before I talk of absorption, I have to tell you one more thing. The inner lining of your small intestine is thrown into villi. So many folds and deep folds. The purpose is to increase the surface area. Why we are increasing surface area? For the purpose of absorption. Why you did not have that many villi in a stomach? Because in a stomach you had to only digest the food. Here you have to absorb. In these villi you have blood supply. Of course, you have blood supply in all parts of your body, but because surface area is more, you have more blood supply. This blood or blood vessels are going to absorb this end product of protein digestion, amino acids, and also end product of starch and carbohydrate digestion, that is glucose, will be absorbed by blood. Coming to fat, fat molecule even in the form of monoglyceride is very big. Blood vessels cannot absorb it. It is absorbed by lactase, and finally, it is given to the blood vessels and it will reach the cells where the assimilation will take place. So, with this we come to the end of this topic, and I am sure you have understood how succus entericus is acting in the small intestine, how digestion in the small intestine is taking place, and how small intestine is helping in absorption of food. We also discussed about nucleases, which will act on nucleic acids and digest it to nucleotides and then to nucleosides. Now, nucleosides can be absorbed. Absorption will take place in the small intestine, and details we have already discussed. So, with this we come to the end of this session, where you have understood digestion in the small intestine, the absorption and the importance of absorption and the importance of villi in the small intestine. You have also understood that absorption of proteins and carbohydrates in the form of amino acids and glucose will be by blood vessels, and absorption of fat will be by lacteals. I hope things are clear to you. With this, we come to the end. Thank you.